Hi, I'm Alicia Tivoli, and today I'm going to teach you all about restriction enzymes. First, let's start with the background of DNA. DNA is the genetic material inside cells that create the fundamental and distinctive characteristics of organisms. In its most basic form, DNA is like a ladder, with phospho-sugar backbone and nitrogen bases making up the rungs. Adenine always pairs with thymine, and guanine always pairs with cytosine. This DNA sequence can also be written like this: T, A, C, G, etc. Now onto the restriction enzymes. Restriction enzymes come from bacteria, and they are bacteria's immune system, where the bacteria uses the restriction enzymes to cut up a virus's DNA into smaller, non-infectious fragments. Scientists have learned how to extract these restriction enzymes and use them for human DNA and DNA analysis. Restriction enzymes work by breaking the phosphodiester bonds in the backbone, which then create a chain reaction to break the hydrogen bonds between the bases. Restriction enzymes can be done in a blunt cut or a sticky cut. A blunt cut cuts straight through the whole pair, whereas a sticky cut only cuts one from two pairs, like this, creating a segment that looks like this, where these bases want to find the complements. Some applications of restriction enzymes include crime scene forensics, evolutionary relationships, gene detection, maternity and paternity identification. All of these use gel electrophoresis to separate DNA fragments by size. In this example, there are 3 active sites on the DNA at 6, 18, and 38 base pairs, creating segments of 6, 12, 20, and 4 base pairs. On a gel electrophoresis, these segments will line up from smallest to largest, since the smaller fragments can travel farther in the gel, creating this gel. Paternity or maternity tests, a more real-life application, we would have the DNA of the child, the mother, and the possible fathers. If the DNA is accounted for by the mother, then it does not need to be accounted for by the father. However, all of the DNA does need to be accounted for. This DNA is accounted for by the mom, as is this, same with this, and also this. This fragment, however, is not accounted for by dad 1, but it is accounted for by dad 2, proving that dad 2 is a genetic father. This is further supported by this DNA sequence that is also only supported by father 2.